189 of the Stem Cell Podcast, Heart-Forming Organoids, with Dr. Robert Zweigerdt. Hello, everybody. This is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. This podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying this listen, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Robert Zweigerdt. He's from Hannover Medical School. He's on the podcast today to talk about his research on heart development and modeling. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, ISSCR 2025 is coming up in Hong Kong. Late-breaking abstract submission is open until March 19th, so get your abstracts in now. Check out the deadlines and the program at isscr2025.org. And also, just a heads up, we have season 2 of our Lab Coats and Life podcast coming out on March 5th, so keep an eye on that too. We're going to start things off with a dive into a Nature Communications paper that's intersecting a couple of exciting pieces of technology that we've alluded to here on the show. One is, of course, embryo models, perhaps the hottest subtopic in stem cell biology right now, but intersecting that with AI, machine learning based approaches to predict the best embryo models and perhaps the best way to create these embryo models. And this is work that's coming from none other than Magdalena Zernica-Goetz's lab, and first author here is Paolo Caldarelli. Yeah, so the title here is AI-based approach to dissect the variability of mouse stem cell derived embryo models. And just to start things off right there, this is mouse focused, and of course there have been mouse embryo models emerging for the last few years now, and certainly human as well. I'm sure that the next phase of this study is going to look at human embryo models. And indeed, like we've talked about, these have been cutting edge pieces of technology that has really taken the stem cell field by storm. They've, in their words, transformed developmental biology, opening up that black box, right, offering insights into embryogenesis without the constraints of natural embryos. And in particular, this is exciting for human development. And since we haven't had great model systems to study that black box of human development for ethical, for technical reasons, and so on. So these models have really taken over the field. But there's variability with these models and really any stem cell model out there in terms of how they develop. Standardizing them is a big concern as well. Reproducibility, high throughput screening. You want to improve on that variability that's inherent with these systems. So to address this, they developed a deep learning model to enhance the reproducibility of selecting certain stem cell derived embryo models. So which ones are the best for modeling development and so on. And so to train that AI data set, they used live imaging and classified 900 mouse post-implantation stem cell derived embryo-like structures, or they call them ETIX embryos, into normal and what they called abnormal categories. And again, this is an imaging processing based characterization to train their AI model. And they saw that the best performing model could actually achieve around 88% accuracy at predicting normal versus abnormal embryos at 90 hours post cell seeding, and 65% accuracy at the initial cell seeding stage. And the hope here, again, is to forecast developmental trajectories, predict which one of these models is going to work and it's going to be normal versus the abnormal trajectory. They took their analysis a little bit further and showed that the normally developed ETIX mouse embryos have a higher cell count and also distinct morphological features, such as a larger size, a more compact shape, and they look more, quote, normal. Then they wrap things up with some perturbation experiments that actually showed increasing initial cell numbers, further supporting this finding by improving the normal developmental outcomes. So they, tweaked the initial cell numbers and saw how that you know, characterize the downstream and how that impacted the downstream development of the embryo. So it's a study in the realm of things that we've described before, using AI to predict the trajectories of different developmental timelines. We've talked about AI in the context of like looking at iPS production, iPS differentiation, and predicting which iPS lines might be able to differentiate the best into certain differentiate into certain lineages. I forget the paper that we covered on this topic. I think it was a few months ago, but hey, everybody's starting to use AI in some context or another. Whether it's inside of the technical parts of the science or helping you analyze the science, it's certainly a useful tool. And here, the tool and the utility is improving embryo model selection. Also, like I said, talking about the normal versus abnormal trajectories of these embryo self organizations. And with the ultimate goal is improving consistency in the evolving field of embryo model developmental biology. We got to make these models more standardized, more amenable to high throughput screening. And hey, maybe AI is the key to that.
Yeah, I agree with you there. The kind of straightforward, I guess, application here is increasing the practicality and scalability, reproducibility of embryo modeling here. I guess starting with the mouse, because I guess they have all the, you know, genetically engineered strains and it's more robust at this point, presumably. But I'm guessing it's moving into human systems. And I think there, the applications are perhaps more broad. It's an idea that's been kicking around in assisted reproduction for, I mean, long, long, long time. Of morphometric analysis and live time-lapse imaging and pre-implantation embryo development in order to predict not just embryo quality, ability to implant, not even predicting the trajectory of that embryo post-implantation, but that as an idea too. And the key difference there, and there, I think that this is still an evolving field. While the idea is old, the AI is kind of catching up, closing the gap. But we're still, I think, a little bit murky on how reliable these things are. And to be clear here, I think that's much greater a challenge, right? Because you're working on embryos with, bright field imaging. You can get multiple planes, but you can't really see kind of any molecular resolution. That was, I think, the real strength of this story is the inputs, right? They had these lineage tracing or whatever, the knock-in mice, where they could see the expression in real time of these key transcription factors, which allowed them to look, much higher resolution, multiple Z planes at single cell resolution, molecular detail. So this is a kind of high powered platform. And even there, I think, mostly I would say because embryogenesis, pre-implantation embryogenesis is this amazingly reliable, reproducible orchestration of molecular and cellular events that is very reliable, even in the human, where it's not as reliable in the mouse. In the mouse, it's like 99 out of 100 times. It just goes right. So yes, I think a lot of the degree of not inaccuracy, but predictive fallibility, I guess call it, would be owing to the embryo models not really accurately capturing those intrinsic capabilities of a bona fide embryo. And to close my long rambling comment here, I think maybe that's an interesting application too, is applying the AI to try and tweak the culture conditions where you could get really something that's more robust and closer to that physiological development. So for mouse, as a kind of a primer to move into like culture systems that may work for IVF. So I think the applications of this are really broad. But as maybe you were alluding to a bit there, I think the reason why it got through is because they were like, oh, let's throw AI at this. It's all the rage these days.
It is indeed all the rage. I mean, there's new scientific subjournals in, like, say, NAJM that are specifically focused on AI. I wouldn't be surprised if there's like a cell AI coming out soon, or maybe there's already out one that I'm just, out of touch right now. But yeah, I mean, hey, it's a tool that we're applying to everything these days, trying to make our lives easier, hopefully. But AI, as we've talked about a lot on this show, is limited based on the initial training data set. So garbage in, garbage out. You have to put in the right data set to appropriately train the AI in this context, looking at the normal versus abnormal embryo development. And like what you alluded to, everything here is visual. So they certainly have these really cool mouse reporter lines that they could use to train the AI for this predictive modeling. But perhaps you can think of ways to take this a step further and incorporate molecular resolution, transcriptomic, all the different omics based analyses into these data sets. But takes things a little bit to a higher level, increases the complexity a bit. I'm sure it's something they're working on, knowing Magdalena and their incredible productivity in this area. So we'll wait and see what happens here.
Oh yeah, it's always wait and see with MZG. It's always something good too at the end of that. We talk about AI here, and one of the most impressive things about AI is how we don't even know how it's doing it, right? They're doing deep math with the AI that comes out with a solution, and humans are just like, huh. And that's about, I don't know, pattern recognition. But there's another thing that humans really can't see, right? And that's we can't appreciate things for the most part over these massive time scales of evolution. It's just impossible for our little pea brains to understand the degree of change that can be enforced over these long time scales. But no, the efforts to understand that process indirectly using all kinds of tools in the history of science has been one of the main drivers of innovation. And of course, we talk on this show a lot about how Evo Devo has been kind of broken open, you could argue, by these stem cell systems. And probably even more so with embryo modeling moving forward. But here I'm talking about a story that takes the Evo Devo angle and incorporating uh, pluripotent stem cells to understand differences in brain development between humans and chimpanzees, right? Because despite the clear phenotypic differences, although my wife might argue I have fewer differences from a chimpanzee than most humans, we're pretty similar at the genetic level. 99% sequence identity. That's consensus sequence identity. And so it's been, poured over for a long while how these differences that are so slim at the genetic level can manifest phenotypically. And it can't be accounted for by protein coding sequences alone, right? There's some kind of epigenomic regulatory element. And part of that has been attributed to what are called these human accelerated regions, right? So these human accelerated regions are HARs that are primarily in that non-coding genome. They're conserved across mammals, but in humans, the HARs, they have a statistically significant excess of mutations, nucleotide substitutions. Don't call them mutations, evolution, call it. And that's suggestive that increase relative to other mammalian species suggests that there was like accelerated evolution at these regions. And a lot of these HARs are enriched at loci associated with brain development. And also key here, which we're going to come back to, neurodevelopmental disorders like autism spectrum disorder and schizophrenia, right? So clearly these HARs are linked to neural development. Um, and they have a lot of potential in their regulatory function to glean some insight into how human brain evolution accounts for these massive differences, not just anatomically, but functionally between chimpanzees and humans, or all non-human primates and humans. And the idea here, which is a story from the group of Yin Shen, who's at UCSF, was to experimentally characterize these HARs. Because while you can get like the genomic sequence and do all kinds of like kind of in silico analysis. To really experimentally characterize them, you got to go get in there, right? You need a system where you can alter them and investigate them in the context of their native chromatin, right? So how they regulate all the other genomes that presumably they're affecting, um, and to like link them to specific genes in that are regulated differentially when you alter those zones, right? So as I said in this story, they looked at this cis-regulatory function of these HARs in using human and chimp induced pluripotent stem cells that were differentiated to excitatory neurons, right? And they focused on these 20 HARs that they came to by a lot of, conservation analysis, etc. Looked at the HARs and then the chimpanzee orthologs, and they use this single cell CRISPR-I approach where they couple CRISPR-I with single cell RNA-seq, right? So they get the actual insult and then they can see within that cell how it affects transcriptomic expression. And they found that there was a lot of diverse outcomes when they messed around with these HARs. Among them, for example, attenuated NPAS3 expression, but specifically the mechanism there was by altering the binding efficiencies and affinities of multiple transcription factors that go to that NPAS promoter region, right? So the HAR alteration messed with the affinity. Also, you could alter like or augment the IPS pluripotency. So in the cells, pluripotency also in the neural differentiation capacity of those IPS derivatives was found when you messed around with this 2X HAR 319 because it specifically upregulated PUM2 gene, right? And then finally, coup de grace here, they show using prime editing, so very targeted, hence Nature Biotech here, a lot of tech here incorporated to show that if you do prime editing on this specific locus, a specific HAR that has multiple variants, you can generate one variant that results in increased SOX2 expressions, SOCS2 expression, to be clear there, and result in increased neurite outgrowth in human neurons. So for me, that kind of closed the loop there, right? So you have the kind of in silico analysis, then you have the on the ground in the cell analysis, and then you show relevance here to perhaps how that could affect the cell behavior and by extension, the neural development and evolution of our big old brain. So there was that like Evo Devo angle I loved about this. Also from a practical level, the neurodevelopmental processes here showing how these HARs regulate neurodevelopment. Implicit there is that like how you could modulate neural cell differentiation or any kind of cell differentiation by focusing on, well, probably focus more neural because these HARs are kind of implicitly neural given the evolutionary difference, but still other cell types too. And then circling back to the disease element, this really got me thinking about how, like, talking about autism, schizophrenia, one, the genetic component and that epigenomic mechanism of neural cell differentiation during development and how that can translate to that kind of neuropathology, perhaps owing to neuroanatomy. And then lastly, like more on like a philosophical level, I got to thinking how like autistic people, especially, autistic spectrum, as well as schizophrenics, for that matter. Those people who perhaps struggle or are not with the condition, but are able to function in society. How maybe they are like the next stage of evolution, Arun, and that's how it's happened over all these epochs. Is that there's been these outliers that then ultimately take over. So watch out for those people you've been underestimating. I mean, watch out for the schizophrenics generally because the behavior can be anomalous. But I think on a more philosophical level, watch out, they're coming.
Yeah, really cool story. I mean, we've talked about this before. This is like one of my favorite applications for IPS that's I think often overlooked, but often leads to some really high profile studies. I remember there's another, I think, UCSF study that used cortical organoids in the same context of modeling human versus chimpanzee divergent evolution or whatever you want to call it. Again, I think looking at non regulatory gene elements. So this is a really cool stuff focusing on these HAR regions, human accelerated regions. It's got the IPS element. You didn't actually even mention the organoid side of it, but this had a sprinkle of organoids, cortical cerebral organoids from chimpanzee versus human for some of the comparisons here. The pluripotency link was really cool. And also talking a little bit about the prime editing. I mean, I remember first talking about that initial prime editing paper. I think they came out from David Liu's lab like 2 years ago. And now it's already being incorporated into these really high profile studies. It's a tool, sure, but it's a reflection on the power of the tool. Is how rapidly that's already been adapted. We talk about Cas9, and one of the amazing things with Cas9 was how rapidly it got adapted by any lab around the world, and hopefully prime editing is on the same boat. I haven't adapted in my lab, but maybe I should be talking to my grad students about it. I don't know.
Yeah, that's a great point. This story has it all and really underscores how the generation time from development of a new application to the insight gleaned from it has kind of contracted, and I think it's a testament to one kind of the more open, and particularly in the case of the Lou Lab, how they've effectively like open sourced this stuff in order to move it into application. And the communication collaboration among scientists, I think, has only gotten better in no small part, hopefully, thanks to this very show. But maybe that's a bit optimistic. We're just trying to entertain you guys.
We do our part. Don't worry. I think we're doing our part here, Dalen. Moving on to a science translational medicine paper. It's one of my favorite journals. I've published there every once in a while. Not every once in a while. We published there once. This is. I wish I published there more than once. This is a paper. Like when I first read the title of this paper, I was like, "Wait a minute, this is coming from Mike Longacre's lab, isn't it?" And I was like, "Yep, that's exactly where it's coming from." Because when I think of scarring, wound healing, and novel large animal approaches to study that, that's the first lab that comes to mind. And yeah, I'm biased. He was on my thesis committee a while back, but this indeed is a Mike Longacre paper coming from Stanford. First author here is Shamik Maskarak. Inhibiting mechanotransduction prevents scarring and yields regeneration in a large animal model. Kind of a broad title. But it's something in the realm of what the Longacre lab has been doing for a long time. And he's actually that rare, very rare breed of surgeon scientist. I mean, the training for science is tough enough as it is. You got to do your grad school, postdoc, all this kind of stuff. And training for surgery is also tedious. You got all these years of general surgery, specialized surgery. And so, shout out to the rare, select few, the proud, the surgeon scientists who are, there's not that many of them. Okay. Mike Longacre is one of the best. He's been at Stanford for many years, and his whole thing for a long time is figuring out scarless wound healing and figuring out the mechanisms for that. So, skin healing after injury can, of course, result in fibrotic scar tissue that's detrimental to skin functions. And of course, there's the cosmetic side of it. No one likes scars. You ideally don't want a scar after getting hurt or after having some kind of surgery. And preclinical models have shown that inhibition of one of my favorite signaling pathways out there, that's YAP, yes, associated protein, can actually result in wound regeneration without scarring in mice. And you know why I like YAP, Dalen, because YAP has these ridiculous phenotypes of hypertrophy and like in all different tissue types. Though the one that comes to mind is the YAP model and then the mouse heart, where I forget exactly what the modulation was, but the downstream result was a mouse heart that had twice the size. It has this amazing proliferative impact, the YAP pathway, if you modulate it right. But YAP in this context, again, results in wound regeneration without scarring. And in this study, they're showing that this specific pathway, the YAP pathway, can be further targeted in a pig model of skin healing, which they call a quote human-like model. And one dose of this YAP inhibitor, verteporfin, was actually sufficient to promote scarless healing in these pigs and also wound regeneration. And then they did the molecular characterization Going beyond just the observation, the single cell sequencing, spatial proteomics characterize the presence of different populations of fibroblasts in the regenerating versus the scarring wounds. And they also validated this in a human foreskin graft model in nude mice. And particularly, they dove a little bit deeper into the mechanism and identified differences in enriched myeloid cell subpopulations after treatment and linked this observation to an increased expression of interleukin 33 or IL 33 in regenerating wounds. So, a really cool study, a very translational study. I think that's what a lot of the Longacre studies are, directly applicable almost immediately to, say, situations of wound healing, perhaps being able to apply this YAP modulating compound to get scarless wound healing. So I think there's a lot of translational potential here, and this is just building on top of the decades of amazing work that's coming from the Longacre lab. So as soon as I saw this title, I was like, "Yep, that's Michael Longacre right there."
There he goes again. And we covered a few of his stories on the show, and this is kind of in that seam. But I love how you let off there with that, the person, because there is such a poetry to to who he is as a scientist, the journey he's taken, and the science that he does, right? The surgeon who leaves no trace. What a beautiful idea. And as though, I get it, it's super tedious, but also it's why these things happen. It's the most beautiful type of scientist, I think, who sees the problem in his work, their work, and moves to change that or address it, when everyone else has just assumed it was part of the job. I mean, talk about scarring, with major surgery, and the surgeon who said, "Why did we need to scar?" I mean, hubris maybe, but not so much, came through. But also, not just speaking to the cosmetic element of it, but molecularly speaking, and we'll come back around to this in my final roundup story, is that while I think the early stories were about the scar and the wound healing, now here I can see how he's digging mechanistically into the cellular basis, the molecular basis, um, to hack it, because aberrant healing, maladaptive fibrosis, it's healing gone wrong, right? So you can imagine how, if you can really pick this apart and get, scarless wound healing, you can get scarless, you know, whatever the pathological element of healing, you can kind of abbreviate, mitigate, whatever. So I think that's a really exciting thing about how this story, how he's taken this all the way down the rabbit hole. And I think it's not just going to be cosmetic and improved healing, of surgical incisions or wounds. I think we're talking about, fibrosis as a target writ large.
Yeah, really good stuff coming from Mike Longacre's lab. And do you remember the other interesting part of his history, his backstory? Do you
Right.
remember?
Yes.
Yeah, yeah. So he is a basketball player
He's a big man.
and he's a big man. Yeah. Well, he was a guard, but way taller than me, that's for sure. And he was a guard on the Michigan State 1977 National Championship basketball team. And one of his teammates happened to be none other than Magic Johnson. He was, of course, an all NBA player for a very, very long time. So I definitely don't want to see him on the court. He'll dunk on me even in his age. But I'm glad that he has taken this shift in his career and focused more on scarless wound healing. Although I'm sure he can really get some buckets even today. So shout out to Longacre, shout out to their great lab, and can't wait to see what's next.
Yeah, well, when you put it that way, I guess when you play a guy who's literally named Magic, nothing seems impossible. So keep at it, Mike. I'll see you on the court one day. I'll take a beating from you just to see if you can get those knees bending. As I said, getting to fibrosis. We're talking today about lung, right here, idiopathic pulmonary fibrosis, which is a major, major problem, progressive, intractable, and as I mentioned, fibrotic, disease, in this case of the lung, with a pretty high prevalence, roughly 1 in 1,000 people worldwide, although there's a range there. And the key here that's so gutting is the median survival and diagnosis is 3 to 4 years. And every year in the US alone, 30,000 people die. So this is a big problem. And more than that, there's just no real clear approach. There's some couple of drugs that can slow disease progression to varying degrees, but the only definitive treatment, as you would guess, is lung transplant. And of course, we know the story there. Not a lot of donors. Even when you can find a lung donor, the 5-year survival is just under 60%. So it's a grim prognosis for those diagnosed with IPF. And what's thought to drive it is, as I was alluding to, is this aberrant regenerative response to injury, whether it's induced by age, environmental exposure, sometimes genetic predisposition. There's genetic disease that has IPF as its basis. But at the cell level, it's due to these type 2 alveolar epithelial cells in the lung alveoli that go wrong. But there's not really a very clear understanding of the mechanism of pathogenesis of IPF, because you wait for it, not a good animal model, right? Because the mice, they got lungs, yeah, but they got very different lung biology. And even other animal models that you might use, the biology of the human lung is unique, with several additional potentially regenerative cell populations that are not in the mouse. And those same cell populations are what you find accumulating in patients with IPF. So it's a problem. And as you know, I was going to say, human pluripotent stem cells present a really unique solution across the board, right? Of course, they're human, but you can model kind of the cellular aberrant behavior in vitro. That's like what these in vitro cell disease models are really good at. Maybe not the architecture or the meta, but in a dish, yeah, you can see how cells change and go wrong. So that was the effort of Hans Willem Snoek, who is at Columbia University. He's been working in the lung and developing protocols for pluripotent stem cell differentiation toward alveolar cell types for many, many years, made many great strides. And this story, they went Rambo, high efficiency here, crazy, it's like almost gloating efficiency, going after these unique cell populations that are in the distal respiratory airways or the terminal and respiratory bronchioles, right? And they generate these protocols using this 3D expandable spheres. They call them induced respiratory airway progenitors, or IRAPs. And these spheres were around 98% made up of these respiratory airway or terminal respiratory bronchial cell types. And this is where it got a little gloating. You can take one pluripotent stem cell and you can make 10 billion IRAP cells. I mean, who needs that ratio? That's insane. But it speaks to how careful and efficient they were. And like when you see numbers like that, you just got to assume, I don't know about assuming, but it suggests, we're working along the physiological differentiation pathway here with very high efficiency and reproducibility. So they also were able to differentiate these progenitor IRAPs into transitional type 2, or like a transitional type 2 like alveolar epithelial cell, and then convert that into a population that looked like these mature AT1 cells, which is part of the differentiation process. Also, what goes wrong in the context of disease, and then, of course, to affirm the capacity of these things to do the disease modeling, they did this genetic deletion of this Hermansky-Pudlak syndrome 1 gene, which, as you could imagine, when you did delete that, it's the basis of that syndrome, which causes pulmonary fibrosis. And they replicate that same aberrant differentiation and show that there's a recruitment of these profibrotic fibroblasts. So this trans differentiation to a fibroblast cell type, and then all those cells recruiting, accumulating. So like that to me is, it's a beautiful story on many fronts, and with the linchpin being that these cells are able to model the cellular basis of disease in a dish, and therefore, as you can imagine, a lot of drugs you can throw at this, and that I think is a major tool in a field and a unmet need, a patient population very underserved. So I think this is a major step forward for them, but also just in terms of like a nice protocol. Uh, Nature Biotech. I should revise. The previous Roundup story I had was Nature. This is Nature Biotech. But a protocol here that a lot of people are going to use to make these IRAPs and go in a lot of different ways. So cool story from the Snook Lab, like always.
Yeah, and it's kind of obvious why this is a Nature Biotech paper. You're generating a new type of expandable sphered organoid kind of thing here that's really scalable, could be very amenable to IPF drug discovery for, pulmonary fibrosis. That's, this is immediately translatable and adaptable by industry and also academic folks who are interested in high throughput screening. 10 to the 10. That took me a second. I was like, wait, that's 10 billion, right? That you're right. I think they're showing off a little bit to go, hey, we can go from one pluripotent stem cell to 10 to the 10 of these IRAP cells. But again, just a reflection on the scalability of the system. And, I've been in biomedical research for 15 plus years. I've never heard of Hermansky-Pudlak syndrome 1. Just comes to show you how many different types of diseases there are out there that cause different things like pulmonary fibrosis. Really cool model system here for scalability, but also for disease modeling. Really great.
Yeah, and I don't recall the lab, but we covered a story that, you know, the obvious application you would think of infinite alveolar cells is, yeah, let's regenerate some lungs. And I remember we covered a story from a big lab that's failing me now. You guys can look in the notes, where that was the story. Taking pluripotent stem cell derived cells, injecting them into the mouse, modeling the fibrotic or other conditions, and leading to repair. So, like, add another, I don't know, few million people to the scope of unmet need there that could be addressed with these cells. So, yeah, infinity cells is a good outcome, and that's what you get when you're working out of the Snook Lab. That's where they don't wake up for less than infinity. But a cool story with some exciting applications moving forward. We'll wait to see how that pans out. About to get into a really great chat with our guy, Robert Zweigerdt, who is a delight. So stay tuned for that. But before we get there, we got a quick message from STEMCELL Technologies. And that's really a question. Do you work with human pluripotent stem cell derived cardiomyocytes? You should use STEMCELL's STEMDiff cardiomyocyte media and supplements to differentiate, enrich, expand, and cryopreserve functional hPSC derived cardiomyocytes. STEMDiff cardiomyocyte media is compatible with human embryonic stem cell and induced pluripotent stem cells, and the resulting cardiomyocytes can be used for disease modeling, drug discovery, and cardiotoxicity screening. Learn more at www.stemcell.com/stemdiff-cardio. All right, you guys, coming at us from Germany on today's episode, we have Dr. Robert Zweigerdt, who is principal investigator at Hannover Medical School. Arun and I have long been big fans of the work coming out of his lab, all about the heart, but also all about a lot of other things that we're going to talk about. In the Zweigerdt lab, they're applying human pluripotent stem cells to work on aspects of developmental biology and regenerative medicine that are focused on the heart, as I alluded to there, as well as other tissues and lineages such as macrophages, So it's a kind of common denominator between Arun and I there. Their projects in the lab include producing human pluripotent stem cell progeny, studying heart development, developing model systems, et cetera, et cetera. We're going to talk about some of it, but no way we can talk about all of it today. But really looking forward to this chat. Robert, thank you so much again for joining us today on the show.
Yeah, hi Dylan, hi Arun. It's a great pleasure really to be on your show and that podcast. As I've mentioned before, it's a fantastic opportunity, not only for me but also for the lab members. They're really extremely proud when you mention that work. Including the work that Lika has initiated on the heart-forming organoids, and that Miriana Dardano has now progressed in the blood-generating heart-forming organoids. So it's a real pleasure. Thank you for having me and having us here in your show.
Yeah, well, the pleasure is all ours. And I'm an IPS cardiomyocyte biologist. I was going to read your papers anyways, but now we get to talk about them with yourself and definitely highlight the folks doing the work. So your lab has, of course, been at the forefront of cardiac biology and pluripotent stem cell biology for a few decades now, with work ranging from scalable suspension culture of iPSCs to improved differentiation of pluripotent stem cells to cardiomyocytes, all stuff I love. And like we talked about, we've covered a ton of your papers on the show. I'm a fellow IPS cardiomyocyte biologist, so I'm always appreciative of great protocols for differentiation and protocols for suspension culture, like what your lab has published. So to get things started, why don't you actually just give us an overview of what your lab is working on right now, just the broad scale of everything, kind of in your own words?
Yeah, I mean, the lab really started, and that was where I have focused my career for decades, was to differentiate efficiently pluripotent stem cells into cardiomyocytes, eventually also with collaboration partners on endothelial cells, fostering regenerative medicine. So the idea is really to generate pure, very well-defined human cardiomyocytes, and together with clinicians develop strategies how to transplant them back into a failing heart, aiming to replace those lost cardiomyocytes. And we have done quite a bit of work there. There is some unpublished work which we have uploaded to bioarchives, where we have generated by our GMP-like differentiation processes in stirred tank bioreactors highly purified cardiomyocytes and implanted them by injection, which is done by professional surgeons into a damaged non-human primate heart, into macaques, and analyzing whether in such an xenogenic model with immune suppression, the human cells will really survive long-term, form proper grafts, and eventually also induce advanced or improved heart function. So that clearly was one direction I was working on, and I had a chance really being in academia, back to companies, and from companies back to academia, which is a little bit of, like maybe a different career path than others, but I really joined it a lot. And that was really kind of an advantage of my career, taking good sides and ideas from both ends. So commercial as well as an academia, where one gives you more freedom, the other maybe gives you more opportunities in terms of technical equipment, and so on. And we're progressing really this work, and that is doable at Hannover Medical School, since we're directly here under the cardiovascular surgical department, where we collaborate with doctors and with surgeons who enable us to really test these cells in preclinical animal models and hopefully in future clinical applications as well. And interestingly, since we were working on trying to optimize these cardiac differentiation processes, always aiming at having pure cardiomyocytes, we noted, and there was a paper, I think it was in 2016, in Nature Communications by my PhD student at that time, Henning Kempf, who noticed that if you modify not only the Wnt pathway modulation by this chiral molecule, which is very well known in the field, which apparently impacts then on Wnt pathway and on cardiomyocyte induction, but if you even keep that constant, but if you modify the amount of pluripotent cells per milliliter culture or whatever, so the concentration of the cells really, you will either end up with proper cardiomyogenesis, or you will fail with cardiomyogenesis, but having definitive endoderm in place.
Mm.
And that suggested to us that there is secreted factors. And so we thought, why not trying to take advantage of that idea and to encapsulate into Matrigel cells and see whether there's gradients of secreted cells, which we have, so to say, proposed, and some of them which we have really discovered, such as Cerberus and others, and see whether if this is in a proper surrounding in matrigel, will form something more complex, including both lineages, which is mesoderm and endoderm, and that means, so to say, a proper patterned mesendoderm. And I think that is really how both research directions are connected. Yeah.
Yeah, I mean, that's a terrific synthesis in a nutshell that really, I think, belies the amazing breadth of your work. You mentioned it, that you in your path, you've gone all over. I think beyond just industry and academia, because there is this kind of space between, I think, that is unique in itself. But yeah, your work runs the gamut. And that's what's so, I think, going to be exciting about this conversation, is that we're going to get all sides. It's not enough for a half hour, but because we got to put some kind of framework in order to it, I want to take it kind of back to the beginning, because at the heart, we're all developmental biologists, right? And the clinical implications of your work are myriad. But let's take it back, because this modeling has had a bit of a renaissance the last couple of years, you know, with the human embryo models, et cetera, and the technology converging on this higher and higher fidelity resolution. So we're able to ask questions and get answers that we haven't been able to before. And you've been, of course, although your work is very much focused on clinical application, as you just referred to, you do have an interest, like all of us, at the fundamental bases for all these structures that we're talking about. So I want to take us back. You talk about the heart-forming organoids, but you got some work that's moving back into kind of the enlage before there, with the mesendoderm and structural differentiation in ways. I'll let you elaborate. I don't want to put words in your mouth, but tell us. Will you mind give us a little sneak preview of that work?
Yeah, that's really super exciting. And Liam Wilson, a highly talented PhD student in my lab, is really performing that type of research. And as you may have seen from the publications, so far we really have focused on endpoint analyses, meaning about day 10 to 14 of the heart-forming and blood-generating heart-forming organoids, where we assess the three-dimensional structure and the lineage composition and function and so on of these cells at that time and the different tissues. However, what we thought is, since we have this highly reproducible, complex three-dimensional structure, why not using and taking advantage of that and studying how these complex patterns of mesendoderm priming, but then also the subdivision of anterior and posterior foregut endoderm, which ends up in different areas of these organoids, with the posterior liver-like cells being placed rather on the outside, whereas the anterior foregut endoderm, which eventually may give rise to lung, that this is in the center. And so therefore we have looked back, and this is what we're currently investigating, is to look by a confocal microscopy and by advanced clearing protocols, really in hundreds of these organoids over time, and looking into, after 24-hour steps, into the individual development from day 0, day 1, 3, 5, and so on onwards. And to study that besides confocal microscopy and flow cytometry, so dissociation, and looking into those different lineages and single-cell RNA-seq, also by spatial transcriptomics, which I think is really a fantastic technology to finally not only observe how lineages evolve, how they eventually migrate in the three-dimensional, space, but also how they will finally interact with each other. So a receptor-ligand interaction, which we hope we will kind of assess by these spatial transcriptomics, which then really allows us to investigate the underlying mechanisms. And finally, to combine that with proper knockout technologies of key transcription factors, knowing to play a role from mouse models in that area, And by that, and really combining proper developmental biology in areas which at this early stages is, to my best knowledge, not really well investigated at all.
Mm. Yeah, so you mentioned how this is building on the existing organoid work that your lab has done over the last few years. Let's actually take it back a step and talk actually more about that 2021 Nature Biotech paper led by Lika Draklis, that amazing organoid paper. It's something that I hope to adapt and am adapting into my own laboratory. And as you know, organoids are all the rage these days. We covered your Nature Biotech paper when it came out. These heart-forming organoids that recapitulate early heart, early foregut development. I mean, we're talking about this. These are really powerful models because they're powerful models of early human cardiogenesis, in contrast to some of the mouse models and other systems out there. But also, and this is near and dear to my heart, they're very amenable to high-throughput screening for drug discovery applications and so on. So if you could you take us back, to those days about the development of this protocol, development of this paper, this Nature Biotech study, how you got there, and maybe about how you can improve these things even more?
Well, it really kind of goes back to what I've mentioned, this Nature Communications paper, where we discovered if you have, let's say, pluripotent aggregates in suspension culture, and you would modulate the amount of cells that you enter in a given media volume, that your differentiation outcome will be very different. And so at that time, of course, the work on organoids using Matrigel for encapsulation, and where the Matrigel is doing this extra, let's say, magic for proper developmental processes. And this is when I have asked Lika, actually starting in her master's thesis, even before her PhD, why not taking our suspension-derived aggregates, placing them into Matrigel, and then applying the Wnt pathway modulation. And this is really was a mind-blowing result when we have used these NKX 2.5 transcription factors, or cardiomyocyte-specific transcription factor at a readout, and found that we will have these proper structured round shapes that we have, like sushi-like rings. I think when I've spoken about that on a Keystone meeting, somebody came up with that description, where we in the end actually found out it's not just a ring-like structure, but it's a beaker-like structure, which we have tried to describe in that paper. That means that you have your whole proper cardiac layer, which is like a beaker enclosing our non-cardiomyogenic, but rather endoderm, including anterior foregut endoderm, including the core. And then we have like kind of the shelf-like structure on the outside, having what we think also plays a very important role is different mesenchymal lineages, which also can be modified in their specification depending on protocol modifications. Mm. And so this is how the work has developed, by seeing an exciting pattern and then trying to analyze and understand this 3D pattern, which essentially initially was just a green ring, which appeared after day 7.5, apparently indicating cardiomyogenesis. But then really via clearing technologies, confocal microscopy and others, try to investigate these lineages in more detail, combined, of course, with flow cytometry and so on. And since you've mentioned screening, that is, of course, very exciting. And we finally hope, since the protocol in principle is compatible with a 96-well screening platform, maybe not much smaller because of the basic size of these organoids of 2 millimeters. But since this pattern formation is so well reproducible, and since we're also working on a multi-lineage, ideally at least a triple lineage transgenic line, for example, expressing in endothelial cells, in cardiomyocytes, and eventually also in mesenchymal cells or in others, a transgene, such that you can ultimately have an automated monitoring of these patterns, such that whatever drug you add, which may modulate these patterns, can be screened in an automatic manner, and that may be for drug development, but also for investigating systematic, let's say, gene modulation technologies, knockouts, overexpression, and so on. And that's something that we're really interested in and working on.
Wow, listening to you, it really underscores how the science we do is really a collection of all these advanced techniques. And as the techniques become more advanced, the insight becomes deeper. And you need that kind of insight to keep pace with the advancing, organoids as a thing. Their sophistication has increased in just a pretty short amount of time. We were just talking to Georgia Quadrado last episode about all these high resolution single cell omics analyses that she levered with a big consortium and applied to neural organoids in order to validate the broad diversity of neuronal subtypes, to prove that they had all, and in the brain there's like a lot. So the technology that you need to validate, and in the same vein, the approaches like assembloids, where you can combine multiple organoids to increase the complexity of the model or the connectivity of the cells within that model. And as you're talking there, I'm seeing how you take all these advanced techniques and you apply them to this observation, right? You had this observation, which everything in science is kind of like serendipitous and observation, and then you just lean in on that and you really squeeze all the juice out of that. I mean, there's still some juice for sure, but you really squeeze hard on that. And what's most amazing to me is the ability of your cardiac forming or heart-forming organoids, to mirror the developmental cardiogenesis. And we talked about that in Nature Biotech, and you just really described it in detail. But the other story you had for me that really drives home, as Arun will tell you, I love developmental hemogenesis, is these blood-producing heart-forming organoids. I mean, what do these organoids not do? We just had, again, call back to an episode before we had a conversation with Elizabeth Ng and Andrew Elefanti about this long and sinuous path to demonstrating the true potency of a pluripotent stem cell-derived hematopoietic stem progenitor, whatever you want to call it. So I have to ask, right? It must have occurred to you with the tech mind how you apply these blood cells. Is it like hematopoietic reconstitution, and you see that as an end game there, or is it more like the role of hematopoietic cells, macrophages, for example, and derivatives in cardiogenesis? Like, whoa, how do you apply these for either biological insight or clinical purposes? Where do you take these heart blood producing, heart forming? Where do you take that next?
Yeah, thank you for describing the amazing work that Andrew Elefanti has and this group has done, and Gordon Keller, of course, and Ditari and others, which is really amazing work. And we're kind of really stepping and by combining our protocol with the advantage or the advancements that has been done in inducing hematopoiesis systematically. And this is really what kind of combining these different protocols and hoping that this kind of makes sense, since the cardiovascular system and the hematopoietic system are developing in parallel. And in parallel means in a timely coordinated and also in a spatially coordinated manner. And that was kind of really the hope that we had, is that we can generate a hybrid protocol which would allow us to keep the Hartanlagen in proper shape, and in addition then having hematopoietic induction. That is really what was the idea that we had and which we have tried to pursue. And you asked about the direction of that work, and of course, regenerative medicine, kind of looking into the possibility also to replace blood, maybe to generate mass production of blood in suspension culture, which we have also collaborated and worked here with as a group on the campus with Nico Lachmann on macrophages and published some work in that direction, is of course a fantastic idea. On the other hand, we feel that our model again is or will have major advantages in investigating proper mechanisms of complex and interactive tissue development and maturation. Yeah. So that means how endothelial to a hematopoietic transition occurs, how hemogenic endothelium is developed, how that relates and how much that develops on proper Hartanlagen and how that can be modified. I think this is where we really see the strengths of that model. And how to progress from here. Again, as you know, there is this embryonic wave and then the transient definitive wave of hematopoiesis, and how complicated it is really to make distinctions between these proper developments. And here Mirjana Dardano is also taking a step that I've kind of tried to explain before, where we look not only into an endpoint analysis, but also on a stepwise development, where we look on how is that hematopoietic differentiation, what stage do we have on day 5 of differentiation, 6, 7, 9, and so on. And by that, trying to make trajectories, meaning at which stage primitive and at which stage transient definitive hematopoiesis occurs, and how these different transient stages that develop along that road, when they are coming up, and whether we will also have a better understanding of the development of these lineages. And then maybe to take it further, as you may know, the next step would be homing of these cells of hematopoietic progenitor cells into the liver. And since we have endodermal induction and at least clear liver Anlagen in fetal liver Anlagen, at least in the heart-forming organoids, where we thought that part of that we may have lost in the blood-generating heart-forming organoid, it appears in yet unpublished work that we can recover that liver Anlagen kind of back or put it in back. And that, of course, would be super exciting to see whether the hematopoietic progenitors can then home into that liver Anlage within the same organoid and then kind of proper progress in terms of maturation. And that's something which we're also looking into and what drives really the interest of Mirjana.
Yeah, we were talking about how these models are becoming more advanced over time. You talked about the hemogenesis potential. Of course, cardiogenesis is something that's foundational to these models. But I mean, it's something that's, I think, also critically important is the scalability of these systems, right? I mean, that's been a long-term goal of the stem cell field, is scalable, cost-effective culture of pluripotent stem cells and their derivatives. I mean, most labs, this may make you cringe, most labs still use that old school method of growing iPSCs and differentiating them in two-dimensional plates and flask, right? But for the purposes of commercial and clinical translation, you gotta think bigger. And you've been at the forefront of this area of study for years now, developing protocols for scalable expansion of pluripotent stem cells and scalable production of cardiomyocytes from pluripotent stem cells. I mean, the dream is for every lab to be able to adapt these scalable differentiation and culture approaches in a cost-effective way. So, I was hoping you could talk a little bit more about the current state of the art in suspension culture and differentiation. And full disclosure here, one of my closest research mentors and colleagues is somebody who you know pretty well, Paul Burridge, iPS cardiomyocyte differentiation extraordinaire, who we've had on the show a while back. I actually worked with him when I was a grad student in Joe Wu's lab. He was a postdoc, and back in that time, about a decade ago, he drilled in my head the importance of these chemically defined serum-free approaches for cell culture, and especially for suspension cell culture. And he's obsessed with refining and simplifying and defining media. So as somebody who's really interested in scalable differentiation, as like a pioneer in this field, could you talk about the importance of these scalable suspension culture approaches, how to bring them to everybody, and also the importance of maybe defined media in these culture approaches?
Yeah, thanks, Aaron, for this question. And Paul, if you're listening, great to hear it here from my end. It's really amazing also. And Paul clearly has inspired us a lot by his cardiac differentiation medium 3 components, which is called CDM3, and so super well known in the field, which really we have used for long term and still do. And just that you're mentioning it, there has been a paper by Niels Griedemann that we have recently published, where Niels was working as a PhD student, and he just completed his PhD thesis a few weeks back, in having a, if you like, fully synthetic medium, so getting completely rid of any type of proteins. And that is kind of what he has achieved by adding some additional components, which are kind of essentially compounds that are known to protect cells from shear stress. And that kind of has really led to a fully protein-free media, which may reduces prices for media by about 25 to 30%. In addition, you get rid of the heterogeneity of some albumin batches, which Paul has always added. And that kind of further simplifies matters. And that has allowed us to work in an affordable manner in a 2-liter scale for in a fully stirred tank bioreactor for cardiomyocyte production. And that clearly is a direction which we're further expanding. There have been some groups which have actually either published or working on even more industry scale production, which is 5-liter, 10-liter. And I'm sure this in future will show us something with multi 10 liters, maybe something in the 50-liter process. That is of really of high and massive interest to us, with the idea to have an allogeneic, if you like, one-fits-all off-the-shelf source for cardiomyocytes. And we have previously spoken about macrophages, eventually maybe also of macrophages and other lineages as well. The other area which interests us a lot, and Aaron, you have mentioned that many people are still working in 2D, where you have plates, flasks, which you have to manually open, with all these risks of contamination, but also with this massive workload that is required, the manual workload which is required, which we think from a commercial perspective and also from a GMP compliance perspective should and must be overcome. And therefore, we think that it is a valuable work and strategy to connect bioreactors and all supportive media that are required and so on, such that you have a closed system manufacturing, meaning that you already cryopreserve your cells, which have ideally been generated already in suspension culture in a bag, which can then be welded via GMP compliant connections to other tubings, such that you can directly fill it without opening any box or flask or whatsoever, and fill your process with that, starting it and having it closed throughout the entire process, which means that the media, dissociation of cells, and so on is done in a closed system. And that actually, if possible and achieved, also simplifies matters and will have a better security of your cells, avoiding any type of contamination, sterility, and that will facilitate and also make processes cheaper and full GMP compliant. And that's kind of actually besides the organoid work, where we're really working in process development, besides the upscaling, but really having closed system manufacturing in place.
Yeah, man. When I listen to you guys who are trying to make this happen, talk about it, it's so fascinating, because it's so clear how deeply you've thought about it and how closely held the feelings are. I mean, I remember in Boston, Arun, when we had that lunch with Paul, and I got him talking about defined media, and he went in. I mean, it was like an hour later, my eyes were watering, but he made his case. I think we got to put you and Paul in like a steel cage with a bioreactor and just see the outcome. I mean, God knows, something good will come out of that, I guess. But what I also love about this whole interview has been, you've really been calling out a lot of names, and that's what this show's about, is acknowledging the players on the ground doing the work. And I think you've done a great job of that. It's clear from listening to you that you recognize that the value of the team and how critical it is to making things work. And I mean, I got to be honest, I was sleeping on the German scientists a little bit. But you know, it's kind of nuts actually that the ISSCR meeting in Hong Kong is just a few months away, because I'm still basking in the afterglow of my trip through Germany for last year's meeting. And in addition to all the great science in the meeting, I learned a lot about how you got like a deep state science in a good way. Deep stable, I should say, not deep state. That's got a negative connotation in the US these days. A deep stable of scientific talent that's really moving the field from within Germany, maybe quietly. And Hannover Medical School, or MHH, as the German is initialized. I'm not going to try and pronounce that. But it's Hannover Medical School is an overachiever among research institutes in Germany, particularly strong in transplantation, stem cell research, regenerative medicine. There's got 30 square kilometers, that's 18 square miles for you Americans, of labs. And as a care facility, takes care of the most seriously ill patients, conducts the most solid organ transplants in all of Germany annually, is the world leading clinic for lung transplantation. So I'm listening. I'm like, wow, this is, did you create the institute, or did you just find the perfect fit? Because this is right up your alley. So no, mystery as to why you're so productive or found a home there. But here's your opportunity to bring some new trainees, because I'll be honest, Germans aren't making enough noise about what great science they're doing, because I didn't know about it and I pay attention. But make your case here. Tell us what makes Hannover Medical School a great place for any stem cell scientist at any level.
Yeah, absolutely. Thanks, Dylan, for giving that opportunity. So first of all, I would really like to thank also Ulrich Martin, who is head of the Leibniz Research Laboratories, which is part of the Hannover Medical School. And also Ajahn Rupavar, who is the head of our institution, so for the Cardiovascular Institute here. Since Uli, at that time, when I was still in Singapore, when I was in my first PI position at an A-Star Institute of Medical Biology, when we met, and actually he was on the trip to an ICCR, which I think at that time was in Australia. And this is how kind of the loop is closed. And Uli asked me, well, you're having a great environment here, but ours in Hanover is even better. And I was like, well, let me take a look at that. And I really found that there is an amazing structure available here. And at that time, Hannover Medical School has received what is called the Excellence Cluster funding from the German government, from the Deutsche Forschungsgemeinschaft, the DFG, which you know very well as a major funding source. And this has been funded for 6 years, for 12 years, which actually really covered at least the first 10 years I came after the first grant was already received. And that really set up a fantastic network of people here in the different areas, which is a lung, heart, and blood. That were the major areas as far as I remember, and we're now reapplying, putting that together. So the stem cell work, which already also was the iPS, well, not only stem cell work, but induced pluripotent stem cell work, where Uli Martin, as I've mentioned, was involved a lot in deriving those from actually peripheral blood-derived cells, which was a very well-cited cell stem cell paper in 2009, where he, I think, was one of the first who was able to reprogram from endothelial cells from peripheral blood. So, and that in mind, again, created here a fantastic network of 25 plus PIs from Hannover Medical School, but also from the independent Leibniz University here in Hanover, and created a fantastic network, which was carrying also my work and which helped me really to settle as a PI here in Hanover. And as you have pointed out, coming back to these opportunities, we on the one hand have really great people working in stem cells, which include pluripotent, but also hematopoietic stem cells, immunology, which is super important. Otherwise, no regenerative understanding immunology is so super important for regenerative medicine. And on the other hand, having the clinicians and the surgeons in the different areas which do full organ transplantation, like in our department, the team of Ariane Lupavar and others can co-transplant heart and lung, which often is required actually for some patients. And that is really the full advantage that we have. And only by that technique, we can translate the ideas that we have for heart repair, because generating cardiomyocytes only and characterizing them is not good enough for developing new strategies, how to retain those in the heart, ensure that they really engraft long-term by proper immune suppression, and finally can really improve heart function, which requires a lot of analytical technologies, as you know, and so on, to analyze the organ, and not only to bring it back, to monitor the arrhythmogenic potential, so the safety aspects, and that kind of is all which is available here at Hannover Medical School in that place. And just to mention and to bring in additional students, maybe, hey guys, you're most welcome to come to us, and we have a number of different PhD programs, and that means international PhD programs. And I would like to emphasize the term international, because just in my group with currently 5 or 6 PhD students, actually, I have people from UK. It was Miriana is from Italy. I had people from India, from South Africa, from Mexico. This is Carlos, actually, who Hernandez, who is now taking over the work or expanding the work that Niels Kriedemann has started. And that's just my group, whereas here on the whole campus, we have more than 50% of international students. And this is also great because it gives a special environment, since the people coming from abroad, they typically feel as a family, whereas the locals, which of course also are super well educated and have fantastic background and knowledge, but still they're kind of, connected also to their families and so on, whereas the international students have the lab as well their home to some degree, which is a most positive way of seeing your work. And I hope that kind of is also really established for some of my students, because this is super important for scientific research, that you need to feel, home and supported by your environment, since you have to take all the challenges of the typical work, as all of us know.
Yeah.
Yeah, 100%. And that, honestly, that's one of my favorite things about modern science, is how international it is. And that's certainly very true about this field. I mean, the ISSCR annual meeting has been in many locations around the world. This year, of course, it's going to be in Hong Kong. Last year it was in Germany, in Hamburg, and we had the great opportunity to be there and meet many folks in the German Stem Cell Network. And just from a personal level, I've been so grateful to your colleagues who have mentored me, who are in the German Stem Cell Network. One person who comes to mind is Sebastian Dika, who also is an iPS extraordinaire. He was a postdoc in Joe Wu's lab also with Paul when I was a grad student there. So it's been great connecting with everyone in Germany and connecting with everybody last year. I actually have a number of trainees who have also trained in Germany. One of my students, Miraj Niazi, who's from Hannover Medical School, it's a training as a cardiologist, and incredible work coming out of the German Stem Cell Network. We actually covered Wolfram Zimmermann's paper recently on the show. Of course, he's at Göttingen with the heart patches. So it's an incredibly accomplished, productive group of scientists, and I'm sure there's going to be more to come in the years to come as well. So thank you so much for joining us here on the show. I was just being a fanboy, honestly, because I love your work. I love cardiac organoids. I'm adapting a number of your protocols into my laboratory. So it's just great to pick your brain a little bit. But before we let you go, we're going to ask you a couple of peripheral questions about yourself. So to start things off, if you could answer any single scientific question, regardless of your expertise or chosen field, what would it be?
Well, I hope it's not getting too philosophical here at this stage, but I mean, I think our field, and you may either agree or having another opinion, but I feel in the end we all work towards a forever life or a prolonged life. Let me put it that way, either by meaning of regenerative medicine, putting in new cells or stem cells, or understanding the mechanisms. And you may also know this fantastic work by Thomas Braun and Johnny Kim, and of course others, reprogramming in situ, at least in the mouse heart, partial reprogramming of cardiomyocytes, and by that, after myocardial infarction, having an expansion of endogenous cells. And you know that Espizo Belmonte has started this in other organs. And in the end, this is where we are going to go, right? But here comes the but, and here comes my question. Once that is achieved, and I'm, also, I feel well, but I'm also growing older, and I would love, like to stay young. How will mankind handle these issues, right? Because that really makes then a difference, rather than nowadays, you may be rich, but still you cannot buy forever life. But there it may be possible, and again, it may sound a little bit too philosophic or too critical, but how to handle that is an important question, and maybe we have to answer that earlier than later.
Yeah, it's the dream for ages, pun intended, I suppose. It's something we've been seeking for a very, very long time now. And we've, like you mentioned Juan Carlos's work with Altos Labs and so on. I think there's a lot of biotech interest in that area as well. So it's not just an academic pursuit anymore. It's a very commercially lucrative area of study. So we'll definitely stay tuned to see what happens there. And finally, the last question, and we love asking this because we get a variety of answers. And I think since this is a show geared towards trainees, I think this is one of the most important things that we ask is, what's the best piece of advice that you've ever been given, professional or not?
Yeah, what I've really learned, maybe it was not one advice, but which I've learned over time, is you need to establish a proper structure and mentoring support to your research group. I think that's absolute key. And what I mean with that is fostering collaborations, steering on the one hand the excitement of people for their project, but on the other hand also bringing in kind of new ideas, and by that excite them even more. And on the other hand, also to make sure that you have a good group spirit. And that is so important in our field, where it is where you have to put in weekend work to ensure that your cells are alive and in good shape. You have to keep each other's back warm. So people have to rely that you're not a concurrent student, which rather would like to see you to fail. But on the other hand, also feel that if I support you, you will support me. And that creates such an important spirit, since there's so many reasons why these experiments may fail, and it takes so much time to achieve something, that such a positive and supportive spirit in the group is really amazing and super important. And I'm trying really to do whatever I can, but it also depends a lot on the personalities that you add to the group, and hope that they will help and support each other. And that also makes my life easy. And there's times, you can believe, when I have to cry, and I'm also happy if they offer me a shoulder to cry on as well. And on the other hand, and that's essentially maybe also which I feel is very important, is while focus is super important, otherwise you cannot complete that project, you cannot complete that paper, and so on. On the other side, on the other hand, being open-minded and looking for new opportunities is super important. And that is which actually helped me to come from that, regenerative medicine, just try to make mass production of cells, towards this organoid field, simply also putting in risks and taking new ideas and say, okay, it may fail, but let's try, let's take time to make an interpretation of these odd patterns that we have, rather than saying, okay, I don't care, I don't want to see the endoderm, I just want to see the cardiomyocytes. The endoderm is bystander cells, which we want get rid of. But say, okay, look, that is interesting, why not taking advantage of it? That's kind of a strategy of not putting all eggs into one basket, and finding a good balance between focus on one topic, but also looking into new opportunities.
Yeah, I mean, that is the central. It's such a hard piece of advice to take because it seems dissonant, right? That you got to stay focused, but also stay open. But that's what we do every day, right? You're looking at something, but we have our peripheral vision. And I think that the real key is taking the science where it leads you, which is another old trope of science, but it's all the same thing, right? Which is that you stay open, as you described, and you follow the answer to the next question and to the next answer and so on a virtuous cycle and loop. That's what you've done, man. That's what you've done. I think that's what you've taught, and I think that's what the greatest scientists at least aim for, is to build on the insight of others and to find the answers within. This has been such a great conversation. You've shared so much, not just about the details of the work, but the way you think. And I think that's what makes this show great for those who listen, is they get a glimpse into that mind of yours. So thank you so much again, Robert, for sharing with us.
Yeah, Dylan, Aaron, it's been amazing, and I really, as you can see and hear from my voice, I really feel kind of very comfortable and chatting and talking to you, and also kind of in the virtual matter, meeting you and talking about our colleagues who helped me really to achieve that and stepping on their work that they have done. So thank you for this great opportunity. I'm really looking forward to progressing that anytime soon and seeing you maybe also at the ISCCR.
100%. We'll see you there. Can't wait to throw a couple back.
Excellent.
All right, you guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at STEMCELL Podcast or via email at info@stemcellpodcast.com with feedback or suggest guests.